All right, welcome to your 5th tutorial, everyone. Okay, today we're going to be talking about passive and facilitated diffusion, different types of membrane transporters, so bacteriorhodopsin, so going over that mechanism, as well as different signaling pathways. All right, just to get you guys into it, we have right off the bat a nice multiple choice question. So I'm just making a poll, but the question is, which of the following statements is correct? Ions and charged molecules require transporters to facilitate their entry into the cell. Glucose, which contains many hydroxyl groups, may pass into the cell through passive diffusion. Oxygen and carbon dioxide pass through the cells by facilitated diffusion through an oxygen pore, and zwitterionic hydrophobic amino acids enter the cell through passive diffusion, but charged amino acids require transporters. Okay, the majority of you guys are choosing A. Okay, so we've got a couple picking some of the other options. So glucose, which contains many hydroxyl groups, remember those groups are going to be polar. So those aren't going to really freely cross the cell, so you do need transporters in order to uptake glucose. Oxygen and carbon dioxide pass through facilitated diffusion through an oxygen pore. So there's no such thing as an oxygen pore. There's like water pores. And then zwitterionic hydrophobic amino acids, if there is any sort of charge, again, that's not going to like passively go through the membrane. And then charged amino acids do require transporters, but even amino acids are going to have some charge to them. Some of the questions or comments said, what is the PSL option? I know students have an exam, so so you know, we can get an idea of how the students that are following along get the right answers. For everyone who is currently studying, just feel free to hit PSL. I kind of understand. Okay. Someone says, by the way, even if a molecule is hydrophobic and it's big, it won't pass through, right? Not necessarily. So that actually opens up a kind of bigger question of, I think someone mentioned this last week, free energies. Depending on the level of hydrophobicity, you can either get these larger molecules kind of going into the membrane. If they're hydrophobic enough, they can stay there, but there's kind of a sweet spot in hydrophobicity that they may come out the other side. But that's like a really big field right now and probably too far past this class. So I wouldn't worry about that. But yeah, if they're too big and bulky, they're not going to pass through. They'll probably get stuck in the membrane if they've kind of reached the level of hydrophobicity to actually insert. Someone says, why is D wrong? So there's no oxygen pore. So I think gases can, oh my gosh, I'm messing this up. I think from the notes, gases actually do pass through. So they don't need pores in order, or it's not a facilitated diffusion for them to go through. Okay. Big and charged. Amazing. Okay. So A was the correct answer. I think most of you hopefully responded. Lovely. Okay. All right, so just kind of briefly going over the types of transporters. So we have passive diffusion. So when we say diffusion, it's just going along your concentration gradient. So we can see here on the outside of the cell, we have a higher concentration of calcium ions and then a lower concentration on the other side of the membrane. So this is going to create a gradient, and the ions are going to want to, through diffusion, kind of level this out and equilibrate. So if the ions are small enough, they can just passively kind of go through the membrane, or if they aren't charged. However, and this doesn't require any energy, these are just going along their gradient. But to make this a faster process, we have facilitated passive diffusion. So this is where you're going to have an actual protein helping these ions or molecules go through. But there's still, if it's passive, moving it down its concentration gradient. So it's trying to equilibrate those molecules and ions. Active transport, on the other hand, is pushing it against the gradient. So it's kind of like creating a gradient. And we're going to see this a lot in metabolism when we look at that proton modem force, where it's pumping out or pushing out protons in order to create that gradient to drive ATP synthesis. And we're going to see that also for bacteriorhodopsin. So this requires energy. If you're moving it against its gradient, you need to input some sort of energy, whereas for the passive and facilitated diffusion, the energy of transport kind of comes from that gradient. So a lot of the times these transporters use ATP as a substrate, and then this is going to drive that conformational change and against its gradient. Now the conformational change is also important because you don't want molecules flowing back. So that's why you kind of get like, they call it like a rocker model. As you see here, it's open on the inside, and then the membrane protein kind of flips and it's open towards the outside. So it's like a kind of rocker model. And this induced conformational change stops molecules or ions from flowing backward. All right. Here we go. Okay, review question 2. How is the rate of movement across a membrane mediated by membrane transporters? What do you guys think? Saturated? Yeah, it is a saturable effect. Maybe think in terms of speed. Someone says rate of flow. Faster, yeah. So this is going to increase the movement of ions. And then, yeah, everyone's saying it increases to a certain point. So definitely this is saturable. Perfect. And then the next question is, rank the different ways a molecule can pass through the membrane in terms of slowest to fastest. So what do we think is the slowest form of slowest? Passive, perfect. Membrane diffusion, yep. And then someone said active transport, excellent. After active transport, what do we have? Then facilitated. And then there's a channel or a pore. Which one do we think would be faster? Someone's saying pore is faster. Yes, excellent. Okay, so if we go through these, simple diffusion is where we're just going to have ions going down their concentration gradient. So this is going to be the slowest form because to get them to cross, it's going to take a while. Active transport is then the next because it requires the binding of ATP, the hydrolysis, and then the conformational change. So there's a lot of steps to happen before you actually get the movement of that ion or molecule across the membrane. Facilitated diffusion by a channel where binding is required. So if you do have to bind the channel, again, this is another step where you get kind of binding and release, even though it's going down its concentration gradient. And this is in more, I want to say, specific channels. And then facilitated diffusion by pore. Pores are usually much larger. So these channels are going to be like fairly specific, like we're going to see in our potassium protein. And whereas you have a pore, it's a lot larger and it's usually a lot more non-specific, so there's not a whole lot of binding involved, so the molecules can just kind of like pass through. So as soon as that pore opens, it's just like very, very fast. Yes, and someone wrote in the comments, pore is usually made from a beta barrel, which is true, so it doesn't require a conformational change. Yeah. So all the other steps require some sort of binding and conformational change, which takes a little bit more time. Someone says, so no binding with a pore. No, not usually. There is going to be like some specificity depending on what the pore is aligned with, but it's not necessarily kind of binding and moving. All right, got lots of questions today. Okay, the rate of indole and glucose transport across a plasma membrane of a previously uncharacterized cell line are shown in the following graph. So the question is, which I guess I didn't put the question, is what kind of transport do you think is facilitating indole and glucose based on their curves? Someone's saying indole is passive or simple diffusion, and then glucose is facilitated. So why do we think that? Why do we think that? Glucose is saturated. Perfect. Right. So our indole is just kind of a linear increase. So it's like the more solute concentration, the more solute that you add, just the faster the rate. So it's probably some sort of passive diffusion or passive simple diffusion. And then glucose, the more you add, you're getting a saturable effect. And someone just wrote, glucose is saturable because all sites are occupied. Perfect. Eventually, yeah, you're going to saturate all the glucose transporters. So you're going to have a facilitated transportation. Nice. All right, I think I saw someone ask what indole is, and that is on the next slide. So we have tryptophan and we have indole. So tryptophan has a similar structure to indole, yet crosses the membrane 1,000 times slower. Why do you think this is? Sorry, a student just asked, does saturable mean that there is a maximum speed that's reached? Yes. So I guess kind of what it means is you're saturating all the proteins. So let's say, okay, I'm just going to go back to the previous question for a second. Let's say at 20 millimolar, we're about halfway. So you've saturated 50% of the pumps that are available. When you get up to about 60 millimolar, that rate starts to plateau. So each individual pump is going to kind of go at its own speed, but when you have all the pumps working together, you're going to kind of reach that Vmax. I guess when we get into more of enzyme kinetics, we're going to go over this. But each one is working at its own max, but if you add in more, those are going to keep pumping, which is going to increase the rate of transportation. But once you saturate, let's say 100% of those pumps, they can't work any faster. So even if you add more substrate in, like more glucose, there's going to be no pump to transport it, and then that's why that rate's plateauing. So the saturable is more referring to the kind of ratio of solute to protein. Someone saying is active transport saturable? Yeah. Okay, so students are responding to the second question. So they're saying that tryptophan is more polar. So therefore it's going to not want to cross the membrane. Excellent. Yeah, so these R groups are the same, or the indole is the R group of the tryptophan. But the tryptophan has our amine and our carboxyl, and that's going to be charged at physiological pH. So the polar amino acid is not going to want to cross that hydrophobic bilayer, as this is going to be really energetically unfavorable. All right. Today's a shorter lecture. I feel like we're flying through, so if you guys do have any questions, please feel free to stop me at any point. All right. Okay, so we went over some of the passive diffusion, so now we're going to get into the more of the facilitated. Someone just wrote wait. Okay. Someone said, do we have to memorize the potassium ion channel mutations and which ions they correspond to? Mutations, like mutations in the loop? Someone want to go back to the previous slide. Or was there, did I miss something that was mentioned in lecture? Okay. Oh, they showed how, okay. Okay, yeah, I see what you guys are saying now. Yeah, I don't know if you need to know the exact residues, but more so like how that selectivity is achieved. So why, I guess, is the difference in residues going to choose the potassium ion over the sodium ion, which we're going to go over in a second. Okay, so potassium ion is kind of one example of selectivity, but there's a range of selectivity in these facilitated membrane protein transporters. So you can have ones that are not very specific at all. So there's a lot of drug efflux pumps, which I work with, can pump out a range of antibiotics, and they usually have a less selective kind of binding pocket as we compare to the potassium ion. Or you can get membrane transporters like the potassium ion channel that is extremely specific to the potassium ion. So they're not all going to have this level of specificity, so you can kind of have a range. So in terms of the potassium ion, as you guys mentioned, we have this reentrant loop, and the amino acids in it are critical for the transport of just the potassium. So the one thing to note is it's the carbonyl backbone from most of these amino acids that's contributing to the coordination of these ions. And then we also have the side chain of the threonine that is coordinating it. So I guess to answer your question of how, I guess, the differences between these different loops is going to pertain to the specificity. So for the potassium ion, usually it's going to be coordinated or hydrated by these water molecules. So this is going to have a kind of water shell around it. And the carbonyl backbone, or the, I guess, the hydroxyl and the threonine, is going to be in the kind of perfect orientation to mimic hydration shell. So when the potassium ion enters into the channel, it's going to have the same type of coordination. If we compare that to the sodium ion, you can see that the water molecules are a lot closer to the sodium because it's slightly smaller. Therefore, when this is desolvated and enters into the channel, you're lacking all that coordination. So this is extremely energetically unfavorable because it doesn't want to strip those water molecules, and that's kind of how you get the specificity for the potassium ion. And then I'm assuming for the amino acids that are specific for sodium, those are going to have a slightly smaller pocket so that it is able to coordinate the sodium. Someone says, can we go back? Of course. Do you have a question on this slide? One more thing to note is, so this is just showing kind of a cross-section of the protein. So there's two more. So this is a tetramer. So there's going to be the four carbonyls that are going to be kind of coordinating this, or the 4 hydroxyl groups. It's also important to note that the potassium ions are going to be in every other position. So we are going to get a water molecule coming in. Otherwise, you would get some repulsion in that channel. All right, a student just asked, can you say again why the sodium is energetically unfavorable? Okay, if we compare the potassium ion in the channel to the sodium ion in the channel, you can see that the oxygens, all 4 oxygens, are able to stabilize the potassium ion. And they're in the same conformation as the oxygens in the water molecule. That's kind of coordinating this molecule. if we compare that to the sodium, you're losing two kind of interactions with the water molecules when you compare it to in the channel. So it's the ions too far away from the carbonyls to kind of successfully coordinate and have that stabilized. So it's going to be less stable than it is when it's surrounded by all the water molecules. So it's not going to be favorable for it to actually go into the channel and lose these water molecules in this extra stabilization. Someone say, would you get something bigger than potassium? Or is that not normally seen? I think that if you go an ion like larger than potassium, I almost want to say that also wouldn't fit because then it would be like too big. So if you can kind of see the size of like the sodium, the water molecules are closer together, and then they get farther apart with the potassium, and then they would get even farther apart with a larger ion. So I feel like that wouldn't properly fit into the channel. So again, this is all showing how the specific size of this pore is what is able to select that one very specific ion. So it's kind of pretty impressive that nature is able to come up with this. All right, any other questions or? Okay, if there are no questions, we got another question for you. Which statement about the potassium ion channel is correct? I will restart the poll. The potassium ion is bound to the OH groups of the threonine and serine residues in the loop. Potassium channel is comprised of 4 subunits, each which span the membrane once. Ion selectivity is enforced by the size of the pore. The potassium channel preferentially binds potassium ions, but also may transport chloride ions, which is correct. Someone has their hand up. Kevin? I don't really know how to respond to that. If you have a question, let me know. Oh, I thought you were asking a question. I'm like, yes. No worries. Yeah, do you guys not see the poll? There's a poll up. Maybe if you, I don't know if you guys can hide it yourself. If you click on the poll icon at the bottom. Okay, we have a bit of a tie for B and C right now. All right, for those of you saying C, or sorry, E, go back to the previous slide. What do you notice about the structure? Yes, so it says, does it span more than once? Yes. So there is actually 2 transmembrane segments. So it's going to span the membrane twice. So you have a loop connecting those 2 alpha helices, as well as that kind of re-entrant loop that's coming out from one of the third kind of shorter alpha helix. Someone says, is ion selectivity not enforced by the amino acids of the selectivity filter? I guess, okay, I see your point. So you're saying C might not be correct because it is specific to that threonine in the selectivity filter. I guess that is true, but it's also the size of the pore is what's not allowing the sodium ions to be able to bind in the pore. So it's like slightly too big. Therefore, if it was slightly more size, they would be able to bind. Yeah, so someone says might be mistaken, but the different amino acids are going to affect the size of the pore. Yeah, so I guess if you're saying like those mutations are going to select now sodium, it's going to make that pore slightly smaller so that those can all be coordinated. Someone says, does the size of the pore refer to the coordination which occurs in the selectivity filter? I guess, yes, so the size is going to be like the actual physical space, but if you have a smaller pore size, you're going to actually be able to coordinate a smaller ion and get all those kind of like 4 coordination points. Or if it's too big, you're not going to be able to fit those. So they're kind of hand in hand. So I'll put the right answer up. Someone says, why is sodium too big? No, sodium is a smaller ion, so the pore is too big to coordinate the sodium molecule. Yes, someone says lower is bigger on the periodic table. Yeah, I think it has another, oh geez, I haven't done some of this chemistry for a while, another valence shell. So it's slightly bigger, has more electrons and protons. Someone says, how many times does it spin the membrane? It spins it twice. Just a reminder, so A isn't correct because it's actually the backbone residues of the serines in the reentrant loop, and then the hydroxyl from threonine, and then this isn't going to transport chloride because it's negative. Someone says, would the potassium pore allow the sodium through because it's big enough for both, or would sodium not bind at all? Oh, nice. Okay, someone responded. It wouldn't bind because it's more stable when coordinated with water than the channel. Yes, great explanation. So the binding in the channel is going to be unfavorable, and it's not going to have all the coordinates. Someone says, can you repeat why A was wrong? So A is incorrect because it's not the hydroxyl group on the serine. It's the carbonyl backbone. So you're going to have 4 of those carbonyl backbones and then the hydroxyl from the threonine. Okay. Great. So this is just one example of how these transporters achieve this specificity, but this is going to be unique to every membrane transporter. And as someone said again, membrane transporters are cool and interesting. So they have, each one of these is going to be very specific to their what molecules they're transporting. All right. The type of membrane transporter is our bacteriorhodopsin. So this is going to have 7 segments, TM segments, and they're going to have their connecting loops. The significance is it's going to be pumping protons when it's oxygen starved. So it's going to try and help create that proton motive force when there's no oxygen. So this is going to stop oxidative phosphorylation, which we're going to talk about in metabolism. So it's instead going to create the proton motive force so that you can still get ATP production. The unique thing about the bacteriorhodopsin is that it has a Schiff base. So a covalent attachment to the lysine, and that's going to be about midway through this bacteriorhodopsin. Someone says 8 TMs? I'm confused. I don't know if we're still talking about the potassium or bacteriorhodopsin. Okay, sorry. So it's light activated to activate the shift base. How is this light activated? So if we look at our shift base here, which is covalently bound to our lysine, it initially exists in this trans conformation. Upon light activation, you get the conversion to the cis isomer. So it's a very subtle change in conformation. So you're just going from trans to cis, but this change in conformation kind of pulls in the alpha that's attached to that lysine just a little bit. So it shifts it in, and the slight change makes a pretty big effect for the protein, which is really cool. So that slight change is going to change the local environment around some amino acids, notably our aspartic acid, our glutamic acid, and it's going to shift the pH. So if you remember, pKa. So if you remember from our amino acids, the local environment is going to affect the pKa, and it's going to make them more likely to donate their protons. So when they're more likely to donate their protons is when you get that proton hopping. So you get our aspartic acid, which is going to kind of release a proton and start this off. So one thing to note with this kind of proton pumping mechanism is that you're not actually like our potassium ion taking a potassium from one side of the membrane and moving it to the other. It's almost like a pinball kind of mechanism where you get one amino acid, that side chain is releasing the proton, bumps it to the next, and then each one gets bumped down. And then you get the release of the proton at the end. So it's not one single proton going all the way through, but this kind of pushing. Also to note, so we have our side chains from aspartic acid and glutamic acid that are participating with the shift base too, but you also have water molecules within this channel. So this is showing like 1, 2, 3, 4. But I think there's a lot more. You guys don't need to know that what all those residues are, but just know that it's the coordination of those side chains plus water molecules that are helping with this. Someone said it's like a hot potato. Yes, kind of, but it's like if everyone had a hot potato and then they all pass the potato at the same time. All right, I'm gonna give you guys a second to kind of just do a little bit of review, and we'll do a fill in the blanks all together. So I'll give you guys a moment to fill that out. The next slide is also fill in the blanks, so if you guys want to go ahead and do that one too, and we can do them all at the same time. That's a good question. Someone says, what would promote the move of protons in one direction? So it's actually pumping protons against its proton gradient. The first movement is actually halfway through the bacteriorhodopsin. So that slight conformational change in that shift base that pulls the alpha helix inward, the first kind of hop is halfway down. So when it kind of moves there, then it's likely to give up its proton to the next amino acid, which is, I guess, going towards the extracellular side. So then it kind of creates this proton hopping from that initial step. So I think it's the fact that the first proton movement is going in that direction that forces that direction. That kind of makes sense. So it's not taking from the intracellular side and moving it inward against the gradient. It's kind of like initiating it right at that position. That helps. Yeah, it's kind of neat how it's able to do that. Okay. All right, we'll move along with the fill in the blanks. So Rebectesiorhodopsin has a blank membrane spanning alpha helices. Okay, someone just wrote them all in. Excellent. 7, lyse 26 or 216, which is, yeah, shift base, covalent, shift base. So the lyse 216 I actually said is a retinol, so it's the chromophore retinol. No, it's all good. That's the correct covalent modification to the amino acid. So we have the chromophore retinol is covalently attached to a shift base. Running through the proton is a series of blank and glutamic residues and closely associated blank. Aspartic acid, awesome. And what are the other molecules? Water. Perfect. Someone was saying, what is a shift base? So our shift base, I'm just going to go back for a second, is this kind of retinol group. And the shift base is this covalent kind of modification to the lysine. Okay, nice job, guys. Leave that up for a second. And if you guys want, I can start on the next slide of fill in the blanks. Is retinol a form of vitamin A? So does it work like a cofactor? I don't know the first part of that question, if it is a form of vitamin A. I would say it's not a cofactor because it's covalently bound. So it's becoming like a part of that protein, whereas cofactors are usually kind of held in place by the weak interactions and not covalently. No problem. All right. In the dark, the blank is protonated. What is our group? That is in the trans conformation in the dark. So this is prior to our photoisomerization. Retinol, yeah. So our shift base or retinol. Illumination, and I said it's going to photoisomerize the retinol, forcing a subtle conformational change in the protein that alters the distance between the shift base and its neighboring amino acid residues. Interactions with these neighbors is going to blank the pKa of the protonated shift base. Someone said increase. So if we want to donate our proton, we want to, it is going to change it, yeah. If we think about our pKa's, if we want to make it a better acid, do we need a lower or higher pKa? Decrease, yeah. So yeah, we're going to want to decrease it to make it more acidic, therefore more willing to donate its proton. Nearby carboxyl group. So this is kind of what that student was referring to. So we're getting that proton hopping happening halfway through that bacterial redoxin. The base is going to give its proton up to an E bar carboxyl group. This is going to initiate a series of blanks between water molecules in the interior of the protein. Proton hops, I'll give you, yeah, proton hopping, which ends with the blank of a proton to the extracellular side. Release of a proton. Perfect. This is going to generate a blank gradient. Proton gradient, excellent, which flows through ATP synthase, leading to the production of, drumroll, ATP. Lovely. So this mechanism allows halobacteria to survive in oxygen and nutrient deficient conditions. Lovely. Here we go. Fun fact, I guess while you guys are filling that out, bacterial adoption is, okay, there's this type of X-ray crystallography. It's like super cool, where they suspended it in a media where they didn't get quite crystals, but it was like almost in crystal condition. And then they shone a light to activate the bacteriorhodopsin, but then also hit it with the X-ray to try and like get a diffraction pattern to try and like capture almost like a movie of the different conformations of bacteriorhodopsin. So if anyone's interested, that's like a really cool article, and they go like very, very in depth to the bacteriorhodopsin. Someone says photoisomerase is light-induced isomerization, right? Yes. Yeah, so when there's light shine on it, you get that trans to cis conformation. Someone says, can you repeat how bacteriorhodopsin moves protons against the concentration gradient? So it's because it's not kind of the gradient that's pushing these protons down. You're getting the deprotonation of the Schiff base halfway through, and it's protonating the carboxyl that's kind of towards the extracellular side. So then this shift, or this kind of deprotonation is what induces those proton hops in that direction. Someone says, are there water molecules between the amino acids? Yeah. So not all of them, but there is definitely water molecules. I'm not sure the exact kind of trail that these protons go on. But yeah, there's some water molecules scattered between these amino acids for sure. Someone says, how does light induce this change? I'm not 100% sure on the physics behind that. I'm sorry. Got the chemistry for the pKa's, but not quite the physics. All right. Okay, before we go on, someone said, can you explain the cis and trans part? I will do that one more time. Right. This is our Schiff base here. And we see we have this trans conformation. When you get the light-induced isomerization, it's now in the cis. So the carbons are now going to be on the same side, making the distance of that Schiff base smaller and therefore pulling in the alpha helix. So remember, trans is going to be opposite sides and cis is going to be same sides. Okay, practice problem. You perform a mutagenesis experiment on bacteriorhodopsin in which you create a mutant that has an asparagine residue instead of aspartic acid. Do you think this would be functional? We'll do a poll. Here we go. Okay, we got some students saying yes and some saying no. Does anyone want to share why they think it might still be functional or why it may not be functional? Okay. So we have people saying asparagine doesn't have the carboxyl group that's going to be able to take a proton and transport it. And then someone also said the Schiff base could still act as a proton acceptor donor. Okay, so you're saying someone's saying taking away the charged amino acids, aspartic acid is charged. Yes, so the answer is going to be it probably wouldn't be functional because, like you guys said, you're taking away the carboxylic group that's able to accept and kind of donate protons. For the student that said the Schiff base could still donate a proton, that is correct. So you could still get that change in pKa. However, you're taking away the proton acceptor by having the asparagine residue instead of the aspartic acid. So it's likely that it won't be functional because of that. Okay. All right. So as it mentioned, probably not functional because you're losing the ability for this to be protonated or deprotonated. Someone says, would the proton still be slightly attracted to the CO in the asparagine? It would be, I guess, in terms of like a hydrogen bond, but it can't be, you can't transfer a proton onto that carbonyl group, and you need the actual transfer of the protons in order to get that proton hopping. So they have to actually like deprotonate one group and protonate another group. So won't essentially any mutation in the active site make proteins non-functional? Great question. Next question is, what about if you mutated aspartic acid to glutamic acid? Do you guys still think that this would be functional? I'm going to redo the poll. Here we go. Okay. So to answer your question, which most students are responding to, it may still be functional. So it's not a hard yes or a hard no, but glutamic acid can be protonated or deprotonated, so it would still have the ability to move protons, but it's slightly larger, so that structural kind of sterics might affect the movement. Oh, perfect. Yeah, someone just said depends on how much the structure is changed by the extra methyl group, really. Yeah. Awesome answer. So maybe it still has the ability, but the structural change might affect it. In terms of, I guess, speaking more broadly, because you said won't it affect kind of any protein? Yeah, it depends. So if it's a conserved mutation where you have similar properties, you might still get some function. It might be worse. Sometimes they find it, they improve the enzyme by making these mutations. So it can either go, it can go either way. Unless you make a non-conserved mutation, then you probably destroy the function of that enzyme. All right. Okay, I guess before we go into GPCRs, sorry, we'll take a quick 10-minute break, and then we'll come back. To those of you leaving for your PSL class, good luck on your exam, and I will see you next week. Someone said, I remember Dr. Patterson saying that replacing a particular nonpolar amino acid with another similar nonpolar acid makes a particular enzyme non-functional. Yeah, so this is going to be kind of specific to whichever protein you're mutating. So if it is an enzyme that's like very specific to that one amino acid, even if you make a conserved change, it can destroy function. So it's going to be dependent on a particular enzyme. It just added another carbon or something and disabled it. Yeah, so sometimes even though it still has that nonpolar character, sterically it's unfavorable. Yeah, so it's definitely possible, but in some cases you can make that conserved mutation, the enzyme's still going to be fine. So it's really variable. Someone says, do we have to do another poll in 10 minutes and then we can leave? We did a poll at the beginning, a couple polls. So I feel like you could be fine, but if you don't want to risk it, you can stick around for another 10 minutes. I'll put up another poll. Can you please repeat why it might not be possible? I'm assuming you're referring to the glutamic acid not being functional in the bacteriorhodopsin. So obviously in the bacteriorhodopsin, it's very sensitive to small local environmental changes because that small shift in the alpha helix, just from that change from the trans to cis conformation, is going to slightly affect the pKa's enough to get that proton hopping. So if you do mutate it, the aspartic acid with the glutamic acid has an extra carbon group. So it's slightly larger, so it might affect that local environment, and then you're not going to get the kind of proton hopping, so it might not be sterically favorable. So it might possibly not be functional. Sorry, I'm a bit confused to one of these questions. If you change the pH to deprotonate the amine and create the shift of the double bond of the carbonyl to the imine, I think, and to C, then oxygen would be able to bind to proton when it work. Is that for the asparagine residue? Yes. So the thing is, asparagine can't be protonated or deprotonated unless, I guess you're referring to the backbone, but then the backbone's kind of tied up in the peptide bond. So the carbon or the amide on the side chain of the asparagine can't accept a proton or donate it. It may be able to participate in a hydrogen bond, but you're not going to be getting the actual movement of the proton. Yeah. No problem. Ooh. Okay, one sec. I need to get my charger. Hold on. Okay, we're back. Chloe, what was your major in undergrad? I was in biochemistry, and I'm still doing it. Woo. What type of majors do we have in this class? I feel like this is not just biochem. Molecular biology, immunology. Biochem and bioethics, cool. Neuro, chem, physiology, heart and disease. Wow. Political science, shout out, nice. Trooper. PSL, I don't know what PSL is. I just can't not think of. Is it physiology? Economics, nice. Ooh, we got lots of diversity in here. Health studies, psychology, econ, cool. Evolutionary biology, I like that. Nutrition. Do you see someone saying pathology? I took one pathology course in undergrad and I thought it was so cool. Biological physics. So we're doing the biological physics. Can you explain the photoisomerization? Genetics, history, math, nice. Oh, that's cool. Okay, so we've got some big ranges in here. I like it. Here I am, just continuing with biochemistry my whole life. Chloe, how did you know you wanted to do biochem? Ooh, I like originally didn't like chemistry in high school, and I thought it was the last thing I've ever wanted to do. And then I really was interested in medicine, but I didn't want to go the life science route because I didn't want to be a doctor, and I felt like a lot of those courses were tailored towards it. And I went to an info session for biochemistry. It sounded cool. The people in it seemed really chill, and I was kind of like, oh, that's interesting. You know, it's not like super competitive. Everyone seemed really nice, and the material sounded interesting. So I was like, I'll give it a shot, and I ended up really liking it. So I wanted to do something like medicine related, but not actually like work with patients. So it was a nice kind of happy medium for me. And the people were super chill, so it was good. Do you have any tips for doing research in undergrad? Yes. Look up labs, see if anything is interest to you, and just reach out to the professors. I find that um, a lot of the times they're super receptive and they actually are excited when someone takes time to like read up on their work and say, hey, I'm really interested in this, can I help out? So I would recommend reaching out to profs, especially now for potential, or maybe not now, but like around Christmas time, or December for summer student positions. Ooh, someone's in drama. Oh, that's cool. Someone say not competitive, but biochem major is type 3. What's type 3? Is that not a trait? Someone said they loved chem in high school but hated it after first year. Some chem courses in university were hard. They got really, really in depth. Someone says, when's a good time to email? Like reading week? Any time really, if you want to send them an email saying that you were interested in positions. You can do that at any point really. It's honestly probably better to do it before like weeks like that because I feel like during reading week or over the holiday break, you're probably going to even get more emails as a lot of students are going to be doing it then. Oh, someone says type 3 is a very hard program to get into at U of T. Oh, I went to Queens. So when I was talking about competitive, I guess there was a GPA kind of cutoff for it, but I found the people in it weren't super competitive because I find sometimes students do compete with each other in certain programs. So everyone was really there's a nice community I found to the major. So that's I guess more what I meant by competitiveness. Someone says, were there still research opportunities with COVID? I guess it depends on the lab and the research facility. So I'm not sure about U of T. I know the capacity at sick kids right now is reduced, and depending how things go, it may be reduced further. But I do know some labs still have students, so it is possible, but it might be a little bit harder. Someone said I worked in a lab last year at U of T and it's closed. Yeah, as I said, it's going to depend on the lab and where they're working out of, but I would still try. I don't know what primordial soup is. Okay. Oh, someone's saying we're doing another poll. Sorry, you need to study. Yes, it's 12:12. All right, we're going to do a poll and then we're going to get back into it, but this was fun. Okay, poll. Okay, here we go. Here's your poll. For those of you who need to leave, you guys are going to do great. Okay, we are getting into GPCRs and G proteins. All right, G proteins, heterotrimeric. So we have the alpha, beta, and gamma subunits. You guys are all so supportive in the chat. I love this. Okay, this is going to be bound to our GPCR. So the subunits are going to dissociate when they're activated by the receptor binding. So what this looks like is upon the receptor binding, we're going to go over the conformational change in the next slide. You're going to get the kind of dissociation or the subbing out of this GDP, which is in the inactive form, and then that's going to release the beta and gamma subunits, and then you're going to get binding of the GTP. Upon binding of the GTP, this is now going to be active and can carry out its GPA's activity, kind of carrying on, we're going to see activating other enzymes, and then upon inactivation, it's going to bind our beta and gamma subunits, and then reassociate with our GPCR. Okay. Now, how does, unlike our other transporters that are actually taking an ion and moving it across the membrane, how does just the binding of a hormone or some sort of molecule to the receptor actually transduce that signal to the intracellular side of the cell? So this is because major conformational changes are occurring. So even though nothing kind of major is happening on the outside of the cell when you get this receptor binding, in the inactive form is in the gray, but the active form is in green. So you can see we're getting this huge swing on the intracellular side, which is going to promote that DP release. Now this doesn't look like much, but this is actually 14 angstroms. One alpha helix, I don't know if you guys, here's my pointer. This one alpha helix is moving where this tiny red arrow is 14 angstroms. And if we think about it, the membrane itself is 30 angstroms. So we're almost getting the entire movement of half a membrane, just to kind of give you guys some scale of how much of a change this is. So that's kind of how you get this signal transduction, where there's not too much movement on the outside, but we get this large change on the inside. So then this is what promotes the release, that GDP, and then our alpha G protein can then go on and activate our effector enzyme. And it's TM6 that moves. All right, so the overall cascade of the GCPRs, oh my God, I keep saying that backwards, I'm sorry, GPCR, is we have our, for example, our beta-adrenic receptor, and it's going to bind to a hormone. you're going to get that large conformational change of our TMP6, which is going to kind of activate our G protein, which is our transducer. So we're going to get the release of that GDP, and then the binding of our GTP, which is going to activate that alpha G protein. Our effector enzyme, which is going to be activated by our alpha G protein, is going to be our adenyl cyclase, which is going to produce our cyclic AMP. So this is sometimes referred to as our secondary messenger. So then our CAMP can do a huge range of things. We're going to get into this much more in our metabolism unit. But one of the things that it does is activate the protein kinase A. So this is our cytoplasmic effector. So then our protein kinase K has, again, a range of functions which we're going to get into later. One of them being contributing to a stream of kind of other enzymes, which is going to eventually lead to glycogen breakdown. So if you notice from this, we have one molecule binding to our GPCR, and it's going to induce this huge signal cascade, which is going to lead to this glycogen binding from just one molecule. So that's kind of how you get this signal oh my gosh, amplification. I am stuttering today. From just one of those molecules binding. So it's a pretty impressive pathway. So again, this is kind of a general overview. Does anyone have any specific questions? Okay, if you do have any, I will get to those. And we'll do some practice problems for now. So one molecule, true or false. I guess I'll do a poll for this. I guess yes is going to be true, no is false. One molecule of hormone may produce numerous secondary messengers, such as CAMP. Awesome. Most of you guys are getting the correct answer here. So it's true. So you just need one molecule to induce all of this downstream pathway. Do another poll. True or false, activation of G proteins involve a direct interaction between the hormone and the G protein itself. Someone says, is there a particular ratio? How many CAMPs are formed with one hormone? I don't know if there's a ratio or not, because it's going to depend like how long the signal's on for. So how long is your G protein going to be activating that cyclase producing CAMP? So I don't know how long that would be, but just like there's going to be a lot of CAMP produced per one hormone. All right, most of you guys are getting the correct answer, which is false for this. So it's not directly between the hormone and the G protein. You're going through that GPCR, transferring that signal through that large conformational change. And then last but not least, we have the beta-adrenal receptor is a transducer protein that produces CAMP. Okay, we've got a bit of a tie. So I'm going to maybe rephrase that and say the beta-adrenal receptor is a transducer protein that directly produces CAMP. See some people changing their answers after that. Yeah, so someone wrote in the chat, adenylate cyclase produces CAMP, right? Yeah, so that's why I guess I should probably rephrase this to directly produces CAMP. So activation of the GPCR is going to lead to CAMP production, but it's the adenylate cyclase that's actually producing the CAMP directly. So that's false. All right. Explain why one copy of a gene needs to be mutated to cause cancer when it's signaling pathways. Why are RAS proteins frequently seen in 30% of all cancers? Awesome. Yep. So you're saying RAS, GTPase, unable to be turned off, leading to unregulated proliferation. Someone says mutation, constant signaling. Yes. These are all correct. Signaling is still there. It's going to keep on signaling. Perfect. Mutation leads to constant signaling. So you really only need one copy to be mutated in order to get this amplification. That is going to persist. And then isn't able to turn off due to point mutation. Yep, perfect. So this is why this signaling pathway is so powerful. All right. Which one of the following statements is incorrect? Choice A, C, D. Sorry, just making a poll. The alpha G protein associates with the adenylate cyclase, results in production of CAMP, which causes a cellular response to break down glycogen. CAMP is a secondary messenger that is degraded by GTPase. The alpha G subunit in G proteins dissociates from the gamma and beta subunit upon GPCR activation. Alpha G protein is a signal transducer for adenylate cyclase. Okay, while you guys are answering this, quickly go back for a second. Can you guys still see the poll if I put it down on my screen? Like, can you see the poll right now? Yes? Oh, okay, perfect. That makes it easier for me to not have a poll on my screen. Only took me to week 5 to figure that out. Okay, here we go. Okay, for the most part, we have most students responding with B. We have some saying C. So the alpha G protein dissociates from the gamma and beta subunit upon GPCR activation. So when you get that conformational change, you're going to switch out the GDP for GTP in the alpha G protein, and then that's going to cause the dissociation. So the incorrect answer is B. And why is that incorrect? Does anyone know how the CAMP is broken down? Someone goes dot dot dot. Phosphodiesterase, yes. All right. Next, we got another. Someone says I have no clue, but I know it's not GTPase. Yes, because it basically is a GTPase. All right. Review question 4, membrane protein receptors and channels can both lead to changes inside the cell, resulting in signal transduction. Compare and contrast how these membrane proteins mediate the changes that occur across the membrane. So this is a bit of a review of our membrane proteins. So I'll give you guys a second to work through this. I can put these up and then we can compare. They have transmembrane domains, conformational changes, yeah. So we have receptors using secondary messengers. Oh, I like that. I didn't come up with that one. I like that. That's a good addition. Differences, the protein receptors doesn't let outside molecules in, but the channel allows molecules in. Yeah, so the difference between transferring a signal versus transferring an actual protein or ion. Need stimulation. Yep, some of them actually do need activation in order to function. Nice, okay. So some of the ones you've got are polytropic, so you need many amino acids. So these can either cross in alpha helices, like some of you said, but they could also cross in beta sheets in order to cross bilayer. Someone mentioned before, usually pores are beta barrels. They rely on conformational changes in order to mediate a change or transportation across the membrane. But I like this for what the ones said, where you can have the differences in actually transporting the signal to the molecules going through. So again, receptors signaling non-actual molecule. So there is still a conformational change, but it's different. Channels creates a pore that allow ions and small molecules to pass, so they can bind or initiate signal cascades. And then someone said, I think they need to be activated. So these are regulated and gated, but you can also put in some of these need to be activated. Someone said both are selective. Ooh, that's another good point. Yeah, so you can have specificity for your ions and molecules. Nice. And then, yeah, I like the addition of the secondary messenger for differences. So we've talked about our signaling in terms of transporting a molecule or ion across the membrane. We've talked about signaling in terms of receptors, and a conformational change is activating a secondary messenger, transferring the signal. And our last signaling is going to be breakdown of phospholipids. So I'll move ahead, but I will come back to the slide if you guys need more time. So phospholipids themselves can act as signaling molecules. So these are going to be broken down by phospholipases, and these are going to hydrolyze our ester bonds. So depending on which phospholipase is activated and which substrate it produces is going to affect the downstream pathways. So phospholipids ases A1 and 2, so this is our carbon 1 and our carbon 2, and it's going to release the fatty acids. Or we can have phospholipases C and D, and these is going to release either the choline head group or whatever head group it is, and then the head group plus that phosphate. And all of these are going to have different signaling effects. I don't think you need to know the specifics of what each one of those do, but just know that you can get different signals from the different types of substrates. One specific example you guys should be familiar with, though, is the phospholipase A2 that releases arachidonic acid. So this signaling pathway is going to be in response to an injury. So when you get some sort of injury, you're going to get the activation of this phospholipase 2. This is going to release arachidonic acid, which is going to be one of the fatty acid chains. And then this is going to get broken down by this COX enzyme. This is going to then activate these ecosanoids, probably pronouncing that wrong, which is going to then signal for thrombus formation, vasoconstriction, and pain and fever. So if you think about this kind of downstream effect is going to kind of go back and try and help kind of cure this injury. So we're going to get thrombus formation, so obviously you're going to want to get the platelets and kind of clot formation. Vasoconstriction, you're not going to want to get blood flow, and pain and fever, obviously, to try and reduce inflammation. So aspirin actually is going to inhibit the COX enzyme. So if you have a fever or headache, this is a signaling pathway that it actually acts through. It's an injury that breaks off the phospholipid. I think it's the injury that releases the phospholipids. So it's like the damage to the membrane, and then this gets broken down. And then it's saying if there is arachidonic acid kind of like free floating, then it gets down by the COX enzyme, I think. But mind you, I feel like the calcium is also going to be slightly off. So I'm not actually sure the specifics on how this phospholipase 2 is being specifically activated. Two. Oh my gosh. Does the stars. So we're saying our cyclo. Yeah, our COX enzyme is going to break down arachidonic acid, yes, to produce these kind of hormone signaling factors that are going to then contribute to the thrombus and vasoconstriction. Sorry. Oh, I see what you're doing with the stars. Got you. Okay, so the general steps of this pathway I would be familiar with because it usually comes up in exams, but I guess it'll be maybe show up in a quiz. All right. Okay, so we've done a lot with our signaling. So now we're going to look into actual binding. So for example, with our GPCRs, when they're bound to molecule, for instance, or any kind of enzyme, I guess. But just as a reminder, we're going to go over our dissociation constant. So if we kind of look into our enzyme kinetics, we're going to have our enzyme and our substrate. And then we're going to get the association or the binding substrate to our enzyme. When we look at more of our Michaelis-Menten kinetics, we're going to get the conversion of that substrate to our product. But for now, we're just looking at the association of our enzyme and substrate to look at that binding affinity. So our KD is going to be a measure of the acid strength or the binding strength, and it's going to be comparing that on rate to the off rate to see if you do get that kind of substrate enzyme binding, how is it to then release the substrate, or are you going to get that tight binding? So the lower the KD, the stronger the binding interaction. Someone says, what does on rate and off rate mean? So it's just the association of the substrate. So that binding interaction. So it's kind of like the equilibrium constant, whether it's like likely to bind. Yeah. Okay, so that was just a little prep for our review question 6. Upon hormone binding, you wish to determine the hormone binding specificity of the newly identified membrane protein, and you determine the following KD values. Which hormone has the highest binding affinity? And I'll do poll. Awesome. Okay, majority of the students are getting the correct answer. So it's going to be 0.1 micromolar. So this is the lowest KD. If you do get questions like this, do be cognizant of the units. So this is in millimolar, and these are in micromolar. So these are going to be smaller. Someone says, why are answers A and B? Thinking? I'm assuming. So the correct answer is A because it's the smallest value. I think that's what you're asking. I'm not sure. Okay. The next part of this question is, upon hormone binding, sorry, one sec. Upon hormone binding, an increase in intracellular CAMP is detected. What can you conclude about the structure of the membrane protein and the mechanism of its action? Oh, I get what you guys were saying. It's a GPCR. Yes. Someone's saying it's adenylating or activating adenylate cyclase. Perfect. Involves G proteins. Yes. So this is all part of the mechanism of action. CAMP is a secondary messenger. Perfect. And what are we getting the CAMP from? What's the initial substrate that CAMP is produced from? ATP. Awesome. Perfect. So the unknown protein is likely a G protein coupled receptor. So just to go over the mechanism of action, we get that hormone binding, the release and the activation of proteins. This is going to stimulate our adenylate cyclase and produce cyclic AMP from ATP. Awesome. Someone saying what's the difference between adenyl cyclase and adenylate cyclase? I think they're the same thing, or I might have just written that different. I have been using those interchangeably. Sorry about that. Okay, and that's actually it for today because there was one last lecture. So if you guys have any questions, I'll stick around for a bit. I've got 20 minutes left, and if not, you guys have a lovely weekend. Good luck with your exam. And I'll put in a poll just in case anyone missed any. Oh, I see a student saying that they joined the section around noon. Okay, I can stay online and put up a poll later on. Okay, so I'm just searching something up. Okay, yeah, 2 prostanoids. Yes. Okay. Sorry to answer. Someone asked to go over the phospholipase pathway. Okay, so just to go over this one more time. Someone says, is glycolysis and Krebs cycle going to be taught in this course, like in detail? Yes, we're going to be going over that in quite some detail in the metabolism unit. Okay. Oh, I'm going to just stop recording. Oh no! Oh no, I stopped sharing. I'll stop recording. Hold on. Ah.